So now we are going to discuss about hepatitis. That is the last virus that I have promised to discuss in details for now before exam. So hepatitis, as its name suggests, it will going to infect liver cell, because remember we call liver cell as hepatocytes, right? So this is liver cell, and after entering inside the liver cell, it will cause, or it will force the MHC class 1 molecule to present one or a part of its protein. Okay, this part of the protein will be recognized by CD8 plus T cell. Okay, that is activate, or that ultimately promote cytotoxic killing. Okay, we all know the cytotoxic killing. It is the phenomenon provided by TC, right? Cytotoxic T cell. So this cytotoxic killing will promote or will facilitate the apoptosis of that infected cell. Okay, so this is the normal infection mechanism. So first, the hepatitis virus will infect the liver, and it will cause the MHC class 1 molecule to express the protein. Okay, and it will be recognized by the CD8 plus T cell. Okay, that is TC cell, and this TC cell will activate after recognizing it and start the cytotoxic mediated killing, and as a result the cell will be apoptotic and ultimately destroyed, and this destruction result in the liver damage and inflammation in the liver, and that is what hepatitis caused. Okay, so this inflammation is often known as councilman bodies if it is occurred in portal tract and lobules of the liver. Okay, so that is the simple concept, that MHC class 1 molecule will present a part of the hepatitis protein, no matter what kind of hepatitis it is, and it will be recognized by CD8 plus T cell, and CD8 plus T cell will facilitate cytotoxic killing. As a result, cell will be destroyed, and it cause inflammation and pain in the liver. And if it is occurring in the lobules and portal tract, then it will known as councilman bodies. Okay, so what kind of symptoms do we expect from that? So first of all, as it is a liver disease, so we may expect nausea associated with fever, because our immune system is actively participating here along with malice. Okay, so during this inflammation, enlargement of the liver will occur, that is known as hepatomegaly. Along with more and more damage will increase the amount of transaminase in the blood. Okay, transaminase is a special type of enzyme that is used by liver to break down amino acids. Okay, but as liver cells are being destroyed by the hepatitis, okay, hepatitis virus, so there is no one that will use the transaminase enzyme. As a result, the amount of transaminase will be upregulated. Okay, although in our blood circulation the amount of serum amino transaminase is relatively low, but as during hepatitis infection liver cells are not present in normal amount, that is why the amount of amino transaminase will increasing, and it will increase some amino acid transferase protein known as ALT and AST. ALT means alanine aminotransferase, okay, and AST means aspartate aminotransferase. Okay, and those kind of transferase enzyme will be upregulated in presence of hepatitis virus, or when hepatitis virus causing infection, and amount of ALT will be higher than AST in that case. And when the viral load is slowly decreasing, the amount of AST will be decreased first, then ALT. That means when our concentration of ALT will be normalized, we can conclude that yes, that particular individual is fully recovered from that particular hepatitis disease. Okay, so that's what I have explained here. So amount of atypical lymphocyte may also increase, which is known as atypical lymphocytosis. Okay, and after that people will end up with jaundice. That is the ultimate stage. Okay, in acute level. So amount of conjugated bilirubin and unconjugated bilirubin will also associated with that jaundice. So why conjugated bilirubin and unconjugated bilirubin are also associated? Because remember, due to the destruction of hepatocyte, bile ductules are also destroyed, because hepatocyte play an important role in their lining, and as they are destroyed, bile ductules are also destroyed, and which result in the leakage of the conjugated bilirubin. Okay, and as conjugated bilirubins are leaked, the amount of conjugated bilirubin in the blood increasing. Not only that, as hepatocytes are destroyed, that is why the ability to conjugate bilirubin is also decreasing, which result in the formation or the conversion of conjugated bilirubin into water soluble conjugated bilirubin. Okay, so that is why the amount of conjugated bilirubin and unconjugated bilirubin will be increasing. Not only that, this conjugated bilirubin will be filtered by kidney, and as a result dark urine will appear. Okay, so in dark urine may also be appeared during the hepatocytes, and that is the during the hepatitis infection, and that is the mechanism that I have explained here, that will help you to remember or help you to understand why the amount of dark urine is increasing. Not only that, you may find out urobilinogen, that is this one, in the urine. Okay, now let's discuss about what is the fact of urobilinogen. So remember, urobilinogen is the microbial converted product of bilirubin. Okay, that means in the intestinal gut we have lots of microbes, we know that. So those microbes have the ability to convert bilirubin into urobilinogen. Okay, and this urobilinogen in normal case is converted back to bilirubin or bile with the help of liver. Okay, but as in case of hepatitis infection, our liver is absent or liver is damaged, so liver will redirect the urobilinogen in kidney, and kidney will simply facilitate the excretion of the urobilinogen. That is why we observe increased amount of urobilinogen in the urine. Okay, so we may observe increased amount of urobilinogen, we may observe jaundice, we may observe atypical lymphocytosis, we may observe increased amount of alanine transferase and aspartic amino transferase. We may observe enlargement of the enlargement of the liver, that is known as hepatomegaly, and along with some classic symptoms such as fever, nausea, and our malaise. Okay, so acute infection generally lasts for 0 to 6 months, but if the virus along with the infection still persists, it will facilitate the chronic infection. Okay, and during that chronic infection, if in the portal tract the inflammation and fibrosis continues, okay, if during the chronic infection the inflammation and fibrosis continues, it is a detection that our disease is going towards post necrotic cirrhosis. Okay, and also hepatocellular carcinoma, that is known as liver cancer. Okay, so why? because remember, or why we are going to discuss within a minute. I will discuss this after some time, then it will be easy to for you to understand. So now let's discuss about various types of hepatitis virus. Okay, so hepatitis virus is generally divided into hepatitis A, B, C, D, E. So I have prepared a comparative chart that will help you to remember the various properties of hepatitis and various types of hepatitis A virus, hepatitis viruses. Okay, so the first one is hepatitis A virus. So remember, hepatitis A virus resembles with hepatitis E virus, and hepatitis C virus resembles with hepatitis B virus. Okay, and HTB virus is a virus that cannot work in absence of HBV virus. Okay, so that is the idea to remember. Okay, so HIV virus is transmitted through ingestion of contaminated food or ingestion or drinking of contaminated water. Okay, so it may observed in travelers, mainly it may acquired by the travelers mainly. So the transmission route is fecal oral route. Similarly, in HIV you may also find out fecal oral route as the mode of transmission. Okay, if undercooked seafood or contaminated water is ingested. Okay, and in hepatitis hepatitis A virus and hepatitis E virus, both cases you may find out only acute cases. Okay, and now serology. So remember here that in all types of hepatitis virus, two important antibodies are involved, IgG and IgM. In some cases you may find only IgG, and in some cases only IgM, and in some cases both. Okay, in case of hepatitis A and E you will find both them. Okay, so IgG or hepatitis A IgG antibody will help us to determine protective antibody. Okay, that means hepatitis A IgG antibody will help us to determine whether we recovering from the hepatitis or not, whether our patient is recovering from hepatitis or not, or whether the vaccine is showing its action or not. Okay, that means IgG is a good signal, right? And on the other hand, IgM will help us to determine the active infection. Okay, so IgG will help us to determine recovery, and IgM will help us to determine active infection, and same will be in the case of hepatitis E virus. That's why I have told you that HIV and HEV quite resembles with each other, but there is a difference. We can make vaccination in HIV, but we cannot make vaccination in HEV. And remember one thing, HEV is unfortunately dangerous for the pregnant women. Okay, they cause a type of syndrome in pregnant women that is known as fulminant hepatitis. Okay, so that is the difference between HIV and HEV. That HIV vaccination is possible in case of HIV, but HEV you cannot find any kind of vaccination. HEV is dangerous for pregnant women and it causes fulminant hepatitis. Okay, transmission is fecal oral route, acute infection, acute, no chronic. Here HAV IgM antibody will help you to find out active infection, here also the same, and protective antibody will indicate recovery, here also the same. Okay, so now let's move to the HCV and HPV, and you may find also some similarities in HCV and HPV. Just like HCV is transmitted through blood, HPV is also. Okay, transmitted through blood means through during the childbirth, during unprotected sex, during some intravenous drug abuse. Okay, there can be transmission of both hepatitis C and hepatitis B, and in both the hepatitis C and B you may observe acute and chronic infection and acute and chronic infection, but in case of hepatitis B virus, the chronic infection is quite dangerous because the chance of chronic infection is depend on the year. Okay, the smaller the age, the higher the chance of chronic infection. Remember that in case of HBV virus, that means hepatitis B virus. Okay, children with less than 6 years are at high risk. Okay, and children with more less years, that means 1 years 2 years, are also at a more dangerous situation in case of chronic hepatitis B virus. Okay, so here it's in a case of HCV serology, IgG is important. Okay, this IgG is screened with the help of enzyme immunoassay or recombinant immunoblot. Okay, but remember, recombinant immunoblot assay will help us to determine whether a person is infected with HCV or not, more specifically than our enzyme immunoassay. Okay, but unfortunately recombinant immunoblot is not that much sensitive as our enzyme immunoassay is. Okay, that is why we exclude both of these, and we usually prefer HCV RNA test, that is the detection of RNA, or more specifically I should say the detection of cDNA with the help of reverse transcriptase real-time PCR. Okay, if reverse transcriptase real-time PCR detects increased amount of fluorescence due to the primer we have used specific for that particular RNA or particular HCV RNA, we can conclude that the person is infected with HCV. Okay, or if our reverse transcriptase RT-PCR result detect low CT value in case of our sample, we can say that yes, that particular individual is infected with hepatitis C virus. Okay, so I hope you have understand it very clearly. And in case of HBV, the role of serology is quite interesting. Okay, so the role of serology will determine whether hepatitis B will move to the chronic infection or not. Okay, let's see how. So remember in wait a minute. Yes, so remember in HBV virus there are 3 types of serological markers that we want to, or we have to know very carefully. That is HBs antigen, another one is HBe antigen, another one is HBc antigen. Okay, HBs antigen means hepatitis B surface antigen, HBe antigen means hepatitis B E antigen, that is mainly produced by infected cell. Okay, and HBc antigen means hepatitis B core antigen. Okay, and there will be a fight between our IgG antibody and hepatitis B surface antigen. Okay, and IgM will keep fighting with the hepatitis B our core antigen, but main determining fight or main decisive fight is between hepatitis B surface antigen and human immunoglobulin G. Okay, now if our IgG wins, then it is a very good news for us, the chapter is closed. But if hepatitis B surface antigen wins, then the acute infection will be promoted to chronic infection. Okay, so there can be 2 types of chronic infection. One type of chronic infection is non-infective, another one is infective. In case of non-infective, the host will feel quite well because there will be presence of surface antigen, there will be presence of core antibody. Okay, but there will be no increase in the amount of viral DNA. Okay, or viral genome, what you can say. Along with the production of hepatitis B E antigen, in that case will also be not further increasing, because remember, as it is not show any kind of infectivity, infected cell will not also produce hepatitis B E antigen. As a result, the entire process is just like a dormant state, right? So that is why in case of chronic non-infective infection, the host is at low risk, although the symptoms will be contagious, that there is no doubt about that. Okay. And another one is, that is chronic infective hepatitis. That is a very bad news. Okay, because in that case we will still find or still observe inflammation and fibrosis, and that will facilitate post necrotic cirrhosis or hepatocellular carcinoma. Okay, because remember, in that time DNA is continuously replicating, that is why the amount of DNA will be increased, which result in cancer along with post necrosis carcinoma. That I have told you here, right? So I hope you have understood, although I am also going to show you a graph, a diagram about that too. So now let's discuss about hepatitis D virus. Hepatitis D virus will only infect a person if the person is also infected with hepatitis B virus. Okay, they may infect together. If it is occurred, then this kind of infection is known as co-infection. And if hepatitis D virus infect a already infected hepatitis B virus infected person, then this kind of infection is known as super infection, and which is more dangerous. Okay, and remember one thing here, IgG IgM both represent the active infection. So in hepatitis A, IgM shows active infection and IgG shows us protective antibody, act as a role of protective antibody. In hepatitis E, the thing is same. In hepatitis C, we are mainly relying on IgG, that will help us to screen the condition with the help of enzyme immunoassay or recombinant immunoblot technique. Recombinant immunoblot assay is obviously specific but not sensitive, that is why we are relying on RT-PCR. And in case of HBV, there is a tug of war between hepatitis B surface antigen and hepatitis B core antigen and hepatitis B E antigen in one side, along with IgG IgM in another side. Okay, and in the last one, IgG IgM will show active infection. Okay, so let's discuss about it one another time. So hepatitis A, hepatitis E virus. So if hepatitis A virus, in case of hepatitis A virus, vaccination is possible, but in hepatitis B virus, no vaccination is possible. In case of hepatitis E virus, is more dangerous in case of pregnant women, that can cause acute liver failure in case of those pregnant women, fulminant hepatitis. Okay, and this is the main game between hepatitis B virus in the hepatitis E virus, that is a match between IgG and hepatitis B surface antigen. If IgG wins, then okay, very good. But if hepatitis B surface antigen wins, unfortunately, then chronic viral hepatitis and chronic infective viral hepatitis occurs. Okay, chronic infective viral hepatitis is dangerous because it will facilitate post necrotic cirrhosis and hepatocellular carcinomas. But if chronic viral hepatitis occur, there will be relatively a good news, because no DNA or E antigen is increasing, so hosts are at low risk but remain contagious. Now let's move to the diagnosis part. So as here we are discussing about problem in liver, so we will facilitate or we will use liver biopsy technique. We can also use liver function test and abdominal ultrasound test. This abdominal ultrasound test will help us to find out or detect any kind of enlargement or damage in the lung, sorry, not lung, damage or enlargement in the liver, along with tumor and fluid detection. Okay, that is the three type of detection purpose we may adopt. Come to the treatment part. Acute hepatitis A, B, E usually do not require any kind of treatment. Our immune system have the ability to resolve by its own. Okay, resting in that case is more preferable. But if chronic hepatitis B is in your body, then antiviral medication is preferable. Acute and chronic hepatitis, in both cases, antiviral medication may applied. Vaccination is available in A, B, and D. Okay. And now just try to remember one thing. If your immune system is good and you have vaccinated with hepatitis B virus, so the chance of hepatitis D virus infection is already lowered, right? That is a quite interesting thing, isn't it? So in autoimmune hepatitis treatment, we have to use some immunosuppressor drugs such as corticosteroid to suppress our immune system, because here our immune system is malfunctioning. That is why destruction of hepatocytes occurring, right? So here we need to use some immune suppressors. Okay, so that's it for today, guys. So I hope this video will be helpful to understand about viral hepatitis disease. I have not discussed about the hepatitis structure at all because it is a part of virology, but we are preparing it on the medical microbiology perspective. So I think this video will be helpful to you, and thank you for listening to this class.